So as Martha said, I will certainly do my best to make an argument that while, yes, we have a long way to go for really connecting the brain mechanisms of many of our psychiatric disorders and the brain mechanisms of behavior, I feel like we're on a good track, and I certainly feel that fear and threat-related disorders are some of the lowest hanging fruit, if you will, in our field. And I'll try to share with you today a whole range of perspectives on post-traumatic stress disorder, from a clinical to large-scale genomic studies, large-scale clinical studies, as well as how we model these systems and some of the threat processes in rodent studies for discovery to identify novel approaches to intervention and treatment. So hopefully it'll be interesting for you, and I've got about 40 minutes of slides, and then we'll have plenty of time to talk. So to put us all on the same page from the beginning, this is what DSM-5 currently defines as post-traumatic stress disorder. One has had a life-threatening trauma, and then after that, again, the first 4 weeks or month of these symptoms is called acute stress disorder, and that's recognizes the fact that most people in the very immediate aftermath of a car crash or an interpersonal assault or other trauma are dysregulated, have emotion upset, may be somewhat in shock, may have difficulty sleeping, may have a lot of symptoms. But most people, and again, I'll use the round numbers of about 90% of people, will recover from these symptoms. But about 10% of people after a given trauma will have ongoing symptoms. And after a month of symptoms, we call it post-traumatic stress disorder. And after 3 months, we call it chronic post-traumatic stress disorder. We can talk about in the discussion some of the other variations, late onset PTSD, et cetera, as well as some of the complexities around what kind of trauma. But for focusing today, we'll talk generally about life-threatening trauma, and we'll talk about these 4 sets of criteria of classic intrusions, which are really the flashbacks and nightmares that are so relatively unique to PTSD. I'll show you in a minute, there's a huge amount of comorbidity with other mood and anxiety disorders, with substance abuse disorders, and with other psychiatric disorders. But there's a few components of PTSD that are really unique to PTSD, and that's the sort of reliving, feeling as if you're in the moment of the trauma through the flashbacks and nightmares. And as I get further on to talking about modeling and really understanding the neural circuits, it's these components where we think about the really stamped memory of the trauma that make it so unique, and also may make it more tractable than simply a series of anxiety and mood symptoms. The second is avoidance, and that's really avoiding the people, places, and contexts where that are triggers related to the trauma itself. And we see this in all anxiety disorders, but particularly in PTSD, it's the trauma-specific component of avoidance. And then the main switch between DSM-4 and DSM-5, I guess a decade ago, was this cluster D, negative alterations in mood and cognition. So there was always this component of more emotional numbness in the aftermath of trauma. This was added to really sort of capture the depressive symptoms that are in PTSD. And of course, we see high comorbidity between depression and PTSD, but by itself, post-traumatic stress leads to negative affect, anhedonia, and depressive-like symptoms. Furthermore, there can be a great deal of difficulty concentrating and difficulty with executive functioning in people with severe PTSD. And as we get further along, I'll talk about how essentially the brain is taken over by the fight or flight system, these emotional regulatory systems, and doesn't really have time for this executive function and concentration issues. And then finally, the other component that's similar to intrusions is relatively specific for PTSD is alterations in arousal and activity, particularly increased startle and not being able to sleep due to hyperarousal. And when we talk about the adrenergic activation of the sympathetic nervous system that seems to be so dysregulated in PTSD, that well explains some of these symptoms. But what is not explicitly in DSM, but what every patient tells you and clinicians know, is this sense of never being able to feel safe, always feeling afraid. And that leads us really to the bigger picture of how we understand PTSD is in part a disorder of fear. Before I dive further into the specific components of brain mechanisms and neuromechanisms of PTSD, I'll say a few more words about the clinical perspectives. First, as I mentioned, it's extremely comorbid, and In many ways, I think about trauma almost in psychiatry disorders today, almost like we talked about syphilis in the 1800s as the great mimicker of many other areas of medicine. And in many ways, trauma is a great mimicker of all sorts of different areas in psychiatry. We know that childhood trauma is one of the biggest risk factors for everything bad in psychiatry, but adult trauma as well can lead to many of these comorbid symptoms. And again, as a clinician, untreated trauma can look like a lot of refractory depression, refractory anxiety, refractory substance abuse if one's not getting at those underlying trauma issues that may be driving some of the other components. The other thing that's interesting about PTSD, of course, all psychiatric disorders often have a brain-body component to it, but PTSD in particular, through its history, has had it. And so I won't talk much formally about the history of PTSD, but in its current nosology, we really started defining PTSD as it is and really recognizing it in psychiatry with DSM-3 and in the 70s and 80s. And it really began after the Vietnam War and people coming back from the Vietnam War, and really the formulation of a whole set of people who were really altered psychologically from the war. That started happening at the same time that a lot of increased discussion about the women's equality movement, and then increased appreciation for untold stories of sexual assault and violence in the 70s. And those two things together really increased the appreciation for trauma in both civilians and in veterans. And then I'll show you about some of the work that we've done and others in the 90s that really increase appreciation for trauma in our at-risk communities and minoritized communities, and how PTSD in our inner cities is almost an epidemic as well. But in terms of the long history of the concept before the modern terminology, it really goes back as long as written history. The ancient Greeks and Romans talked about people who were forever changed after a battle or after a fire, and yet they had no physical obvious injury, yet they were changed in their behavior, in their emotion. And then in the Civil War, in World War I, terms like railroad spinal syndrome, shell shock, soldier's heart, again, really representing sort of the sympathetic hyperactivation of the cardiovascular response, nervous shock, neurocirculatory asthenia, combat fatigue. All of these, we think, really capture the set of symptoms that PTSD has in the body. And similarly, as well as is increasingly understood in disorders like depression and other stress-related disorders, PTSD is very clearly associated with dysregulation of the immune system, as well as cardiovascular dysregulation and metabolic symptom dysregulation. And across those components, PTSD has been shown to be highly associated with cardiovascular disease. We had a recent paper I'm not going to talk about today, but showing also at a genetic level, polygenic risk scores are shared in risk for cardiovascular disease and for PTSD. And it's likely that some of these, come through these shared components of cardiovascular functioning, sympathetic regulation, and metabolic syndrome and other inflammation. And similarly, cancer is increased risk in PTSD. And so my last epidemiological statement will be, again, about, depending on the sample size, but for example, Kessler's the WHO studies suggest about 7 to 10% of the population is at risk for PTSD. Again, on average of those who've been highly traumatized, it's about 10%. We see about a 50% increased risk in women compared to men. And while part of that is probably associated with increased likelihood of physical and sexual violence in women, there seems to be, even when you control for those, some increase in females. And again, epidemiologically, it looks like females have a higher risk of PTSD and depression and anxiety symptoms, and males generally have a higher risk for autism spectrum disorder and psychotic disorder symptoms. And I guess my final comment is, while it's only about 8 to 10% of those who have PTSD in the general population, in highly at-risk populations, it can be as high as 20 to 25%. And that's in part due to the trauma load of increasing and ongoing trauma exposure, both in battle and, as I'll talk about in a little bit, in at-risk communities that suffer from both structural racism and structural components of poverty and trauma and cycles of violence that also increase risk in at-risk communities. all psychiatric disorders really can be thought of as a combination of nature and nurture instead of nature versus nurture, or modern terms, gene and environment interactions. But I would argue that post-traumatic stress is one of the ones that we understand the best about the role of gene and environment interaction for a couple reasons. First of all, by definition, it's the environment that is causing the disorder. It is this environmental trauma happening to you. But increasingly, we're understanding the role of genes, and I'll show you a little bit about some of the work showing that PTSD has about the same genetic heritability as does depression and substance abuse disorder, around 40%, with pretty robust polygenic risk scores and large-scale genetic studies of over 1 million people now with sort of a genetic architecture of PTSD. But how that interacts with the environment is still being understood. I'll make some suggestions as we go, and some of the data that's been most robust so far is how genes involved in the hypothalamic pituitary adrenal stress axis are differentially altered in the presence of developmental trauma and adult trauma, regulating increased sensitivity of these stress systems. So the first brief component that I'll talk about today is some of our work that, really my first work in the area, and that's in our civilian population. So I trained in Boston, but then spent 18 years at Emory in Atlanta. And my basic science work was with Mike Davis. I'll show you a little bit about in mouse, in the rodent amygdala to understand threat and fear. And at that time, I really started to want to understand trauma more, because this seemed like an area in psychiatry that could be particularly translationally relevant in terms of understanding the amygdala and other emotion brain mechanisms of threat as they might pertain to a human disorder of threat. And we knew early on from the field that the amygdala was involved in PTSD as well. But in addition to working at the VA and really starting to understand the effects of military trauma, I trained at Grady Hospital, Grady Memorial Hospital in Atlanta. And Grady's one of the largest remaining, if you will, charity hospitals in the country that serves a large, like 95% Medicaid population. And it really serves folks who can't get care other places. And it's historically been a great training hospital and a great hospital for at-risk civilians in Atlanta, and it's a primarily Black population. So we started first in the Fulton County Mental Health Center. Just as a clinician, I noticed that people were having a lot more trauma than was being recorded. And in the Grady Emergency Department, which is the main level 4 trauma hospital in the country, and in the psychiatric emergency department, we would see a huge amount of comorbidity of everything coming into psychiatry. But what really connected it all was the trauma exposure. And yet we weren't in the 90s talking a whole lot about PTSD. We were looking at depression and substance abuse and bipolar and all the other components. So we started asking, and we started more explicitly looking at trauma, and it was everywhere, of course. And we found that in our outpatient mental health center, something like 25% met criteria for PTSD with a SCID or a CAPS, but only about 5% of them mentioned trauma or PTSD in their chart. And then we moved to the general medical clinics and asked the question of, is this increased rates of PTSD really just a severe mental illness, or is it really at-risk communities in poverty? And so we started interviewing people from the general medical clinics, for pediatrics clinics, OBGYN clinics, who were not explicitly, they weren't seeking psychiatric services, they were just there for general medical care. And 15 years later, or 20 years later, we've now interviewed almost 13,000 people. And so it's one of the largest studies of civilians and PTSD in the country. And what we found, was just amazing. And these numbers are from when it was a few thousand, but they hold up at 13,000. About 40, it's a highly impoverished population. Essentially, everybody's under the poverty line. But the levels of trauma are just astounding. So 40 to 50% know somebody personally who's been murdered. About the same percent have seen police violence. About half had been attacked. About a third had experienced sexual assaults. About a third had experienced childhood maltreatment. And the rates of PTSD, again, were 25 to 30%. So the rates of PTSD were as high as they are in our military populations. And we more recently have added and started to really learn about levels of discrimination and structural racism. In this group, Nagar Fani, Nate Harnett, Sierra Carter, who's now at Georgia State, have done really some fantastic work with some of the leading data sets or papers using the Atlanta population to really start to understand more where structural inequities and both intersect with behavior and symptoms, but also with brain function related to PTSD. So I'm not going to talk a lot about this today, but happy to discuss it more in the discussion section. But it's been, I think, an important area for us, and I think an important area for the field. And while, of course, this is all based in Atlanta, other studies, folks at Temple and Penn in Philadelphia, folks at Wayne State in Detroit, and in Chicago, in New York, in California, we see over and over again similar rates. If you look at impoverished cohorts, you see very high rates of PTSD and unfortunately untreated PTSD. And we are starting to have data that if you can treat the PTSD, you can also both decrease the symptoms, but also decrease the cycles of violence, and that there may be intergenerational components as well. So if we can treat the parents, it can really maybe help with the kids. But so happy to talk about that more, but I'm going to go more into the clinical and science side from here. So just one slide on sort of our current treatments, but again, happy to talk more. So our best empirically validated treatments for PTSD are really the talk therapies. I'll maybe say the bottom first, which is we only have 2 FDA-approved medications for PTSD. They're SSRIs. It's sertraline, trade name Zoloft, and paroxetine, trade name Paxil, and those were FDA-approved over 2 decades ago now, and there hasn't been anything else. There are a number of companies now that are starting to move things forward, and there's a number of very interesting biologically based new drugs that are hopefully will be able to move forward for PTSD indication. But it really shows that a disorder that can affect up to 8 to 10% of the population, yet we only have 2 drugs and they're 20 years old for it. That we have a long way to go, especially as I'll be making the argument that I think this is one of the better understood psychiatric disorders from a neurobiology perspective. Currently, our best treatments are really the psychotherapies, and it's really the trauma-focused therapies that have the best evidence. And most of them, so Edna Foa from of course Penn is really kind of the world leader in understanding prolonged exposure. And I was mentored by and great colleagues with Barbara Rothbaum, who trained with Edna at Emory. And prolonged exposure really builds on this concept of extinction of fear that I'll talk about quite a bit more later in terms of animal models and in memory. But the basic idea is you come into the therapist's office and you talk about your trauma. And you don't want to talk about your trauma because you want to avoid. But if you have a good therapist, they'll help you regulate your emotion. And a lot of the more recent treatments, cognitive processing therapy, STARE, and a full range of cognitive behavior treatments, and EMDR, eye movement desensitization, really they're all about sort of grounding, emotion regulation, so that you can then do the work of the trauma exposure. And as the patient talks about the trauma, they'll say, what's the hot spot? What's the thing that you're, is most bothered by? And starts talking about that trauma, they'll of course have very high levels of anxiety. And part of the issue that I'll talk about later is not allowing the patient to dissociate, but instead stay present to extinguish. If they can do that over the course of 30 minutes or an hour, and then they come back the next time and do it again, they're a little less anxious, the next time a little less anxious, and then you move to the next trauma. And over time, you're essentially retraining the brain to no longer be afraid of those specific events. So in sum, we have medications that are really non-specific symptom reduction, decreasing the general anxiety symptoms, decreasing the depression, and helping sleep, but not getting at the core problem, which is the memory, and that's where the therapies really are best. Good question, Dr. Deere. Are psychotherapies regulated? Not really, and we can talk about that in the discussion. We can talk a lot about how the field does randomized controlled trials with psychotherapies, but that doesn't prevent a lot of people from kind of doing their thing, and that doesn't have the same regulation that FDA medications do. Okay, so moving then on from kind of that broad picture of the epidemiology, current treatment, to where do we go from here? So again, I would like to make the argument that PTSD is tractable, and I'll make the argument that as a shared neural circuitry across species, we know when it starts. It's one of the few psychiatric disorders where by definition we know when it starts. It's at the time of the trauma, and that leads to both us to be able to understand it and follow it chronologically as well as model it. And it involves learning and memory. We often think of psychiatric disorders with learning and memory as really dementia and loss of memory, whereas PTSD we can really think of as a disorder in which you have this overly strong instantiated trauma stamp in your engrams that you can't get rid of and can't get away from. And then you have a memory deficit, if you will, in learning to extinguish or inhibit that fear. So our goal is to have a molecular neurobiology of PTSD leading to predictive markers that lead to novel interventions and treatments. So why do we have fear? So I think everybody is familiar with this general concept of the fight or flight or freeze response. But just to make it a little more deal, so this is a classic slide from Joe Ledoux from almost, I guess, 30 years ago in Scientific American. What it shows a guy walking through the woods and he sees a snake, or maybe it's a stick, but he has a fear response. And what we know is all of our sensory systems are constantly bringing in information from whether it's olfactory or visual or somatosensory. And the first step of that sensory information goes into at least for everything but olfactory, into the thalamus. I mean, olfactory, interestingly, goes directly into the amygdala. And one of the arguments about why the olfactory memories are more emotional than others is maybe that they don't have an amygdala relay. But with the visual system, the eye see this snake-like structure, or maybe it's a stick-like structure. It goes to the thalamus in 1 to 2 synapses. And from the thalamus, information goes to the primary visual cortex, the secondary visual cortex, higher order visual cortex. And it takes about half a second, or about 500 milliseconds, to go multiple synapses until enough of this information is processed that one is A, consciously aware of it, and B, can really process what that information is. In parallel with that, in parallel with the very first visual information, projections from the thalamus go to the amygdala. And then only in 2 synapses in the amygdala, then the amygdala compares this with your lifetime experience of threat memories and threat cues, and activates, essentially in a reflexive way, the hardwired fight or flight system. So that hardwired fight or flight system is activated in about 100 to 200 milliseconds, fully half a second before you're consciously aware of what's going on. Well, that's great for surviving the bear or the lion or the snake. But in the modern world, it leads us to a lot of dysregulated fear in a world that is increasingly threatening. And I think that not only is interesting for our disorders like PTSD, but I think it's interesting for what appears to be an ever-increasing rates of anxiety in our world. As far as understanding it, the human amygdala is very similar to non-human primate amygdala. Cat, dog, rodent amygdala, very similar circuits, and even things like reptiles have an amygdala-like structure. So it's a very old subcortical circuit for survival. To dive a little bit deeper into what we understand now is the basic idea of Pavlovian conditioning, where you have a previously neutral cue, in this case like a green car, is paired with the unconditioned stimulus, the car crash, the sounds, the smoke, the pain, the fear, the threat. And that single trial learning that we now know involves lots of plasticity in the amygdala, that includes NMDA receptors, BDNF receptors, calcium type plasticity. That leads in the future to the conditioned stimulus alone. Now what used to be just a car going down the street or innocuous honk sound now activates all of the same hardwired symptoms as if you were going through the car crash. Essentially, a switch is flipped in the amygdala and previously neutral cues are now activating the threat response of the unconditioned stimulus. And what people like Mike Davis and Mike Fanzelo and Joe Ledoux and their groups figured out in the 70s, 80s, and 90s was hardwired projections from the amygdala, direct neural projections to many subcortical areas, lead to all the same symptoms that we see in a panic attack and the threat-related panic attack: the increased heart rate, the increased blood pressure, the GI distress, the respiratory distress, the increased arousal, the startle response, freezing, and the corticosteroid release from the hypothalamic pituitary adrenal axis. So it's one of the few places in our field where we really can boil down the basic idea of a threat, a learned threat becoming now a conditioned stimulus, activating a reflexive emotional set of symptoms that we see as a panic attack. And simply knowing this can be really helpful to patients, because it can take this "I'm going crazy because all this stuff's happening to me and I can't control it" to, oh, this is what my body does to survive. And now I need to focus on how do I better regulate it upstream, how the prefrontal cortex and hippocampal can better rein these things in. So I'm getting excited about talking, I'm taking too long, so I'm going to speed things up a little bit. So how do we think about this, the broad spectrum? I've sort of laid out for you the problem at an epidemiological level, at a patient level, the basics of the fear system. how do we think about the problem of PTSD and fear disorders? And again, the overall, given a specific fear event, the car crash, the interpersonal assault, the battle, why do 10% of people go on to develop PTSD, but most 90% recover? What are different components? And the field has brought us lots of partial answers. First of all, pre-existing sensitivity. I'll show you a little bit about the genetics data, but again, we know that about 30 to 40% of the differential risk is likely genetic. And then environmental. We know that childhood trauma likely sensitizes these systems. And if you have childhood assaults and abuse, you are much more likely to have PTSD as an adult if you have an adult trauma as well. You sensitize the system. Then we know that the things that happen around the time of the trauma are different. For some people, we know that, for example, interpersonal assault is much more likely to cause PTSD than a sort of neutral car crash, for example. And we know that what happens in the aftermath of PTSD. I'll talk a lot about the concept of consolidation in a minute. Then after people start to have these symptoms, the symptoms themselves can further exacerbate it. So we think that these early acute stress symptoms may differentially lead some people down more to an ongoing sensitization of PTSD and others to more recovery. So what does that look like? Well, the person with PTSD will start to generalize fear. First, they'll say, I was attacked down that dark alley by that person. And initially I was afraid to be in that spot. Then I was kind of afraid to go out at night. And then I kind of started not wanting to be around men. Now I'd really rather just stay in my house. In fact, I'll just stay in my bedroom. The world has become more and more dangerous. And that's in contrast to those who recover. They're able to discriminate emotionally. There are safe places and dangerous places, and they're not avoiding critically, because we know the avoidance feeds into the system and makes you continue to be more afraid. Then I talked about extinction as what we do in psychotherapy, but it also happens naturally all the time. The people who talk about it with their family, have a supportive spouse, relative, family, friends, are able to talk about it and process it on their own pace, are much more likely to have natural extinction in those days and weeks after trauma. Others tend to sensitize. They'll say, Doc, every time I talk about it, it feels like it gets worse and worse. Why would I want to go to therapy to talk about this? And in recent years, again, from Joe Ledoux and Kareem Nader initially, and then many others since then, this concept of reconsolidation, that anytime a new memory is activated, it can either go down this path of being inhibited and weakened, or go down a path of being further strengthened. And we think that this reconsolidation of the memory engrams is part of what leads to the sensitization process. So first of all, by breaking it down into these components, we can study different components and start to break down this complex concept of PTSD. So in the remaining time, I'll just show a little bit about where we are as a field and some of our own work about translational studies of memory consolidation, both in human emergency department studies and following people over time, as well as how that's feeding into our larger scale genetic approaches to understanding the genetic mechanisms of PTSD and targeting some of those pathways. And then I'll end with then talking about how our understanding of inhibiting fear or enhancing fear extinction, could we make the psychotherapy work better by enhancing plasticity to help people to recover faster? So again, the idea of consolidation is that memories don't become permanent immediately. Rather, they initially, in the first seconds and minutes, it's really a receptor, we think, synapse sensitization. That then leads to gene expression changes over minutes to hours. That leads to neural circuit changes, and then you have whole brain regional changes over days to weeks. But the first, so that's the sort of short to long-term memory. Then long-term memories are activated, and they might be, once they're then brought back up in working memory, I'm triggered, they can be reconsolidated or sensitized. I just saw Harshil's question about EMDR. I'll come back to that in the discussion. But in contrast, when that memory is brought back up, they may be faced with opposite data. Okay, this time I'm talking about the trauma and nothing's bad's happening. It's not really happening. I'm starting to learn that the conditioned stimulus is not all it now in this context is not paired with the unconditioned stimulus, and I can learn safety. And that's what extinction, and that's where we think recovery really occurs. But I'm going to talk about for now the idea of consolidation, and this is for two reasons. One, it's one of the best places we can study it, because before all the other complex stuff happens, it's what's happening in these early minutes and hours and days after trauma formation. And more importantly, if we can really understand this and we could target it, might we be able to intervene? And the idea here is right now, if you have arm numbness or chest pain, you go to the hospital and they can diagnose an early stroke or an early heart attack and do some interventions that may very well prevent the long-term sequelae of a heart attack or a stroke. If we understand the biology of early memory consolidation, could we do treatments in the first hours or the first night of sleep that would prevent that trauma memory from ever forming long-term memory, or at least not forming the depth of long-term memory that becomes a trauma? And that could be a great intervention in the emergency department or on the battlefield. So again, Jim McGaw and many others, have laid out the basic idea of these short to long-term memories. In my work with Mike Davis, we started understanding the role of the amygdala and many different gene systems in the amygdala involved in transferring short to long-term memory. But how can we figure out what's happening in humans and start to translate this across species? So about 10, 15 years ago, we started a study between Emory and Miami, Charlie Nimroff's collaborations with our work at Emory, and we enrolled people in emergency departments, again at Grady, right after trauma exposure. And we were able to get blood very early in the early hour or two after trauma exposure. We were able to measure physiological measures, essentially as soon as somebody was medically stable and essentially waiting for the MRI results or the blood test to come back, we were able to interview them if they consented. And then we followed people up for a year and were able to see the trajectory of their trauma experiences as well as the neuroimaging and brain-based mechanisms. The data from here then led to a large multi-site study with initially 14 and then almost 20 emergency departments with almost 5,000 participants over 5 years. It had some hiccups because this was ongoing through COVID, but we still were able to meet most of our milestones. And that was led by Sam McLean, who's an emergency department doc at UNC, as well as Ron Kessler and Kirsten Conan and myself, who are part of different Harvard programs. And there's now a large number of papers starting to roll out of this, but I'll just show you a few pieces of data to make some points. So first of all, what we see in any given patient is in the first weeks, you have a full range of trauma symptoms. This, in this case, was the PTSD symptom scale. We also use the PCL5 PTSD checklist as sort of brief symptom scales. Most people recover. So if you look a year later, most people have very few symptoms. Some have high symptoms, stay high. Others stay low and get a little bit worse. But what was important is, while I told you that we can't diagnose PTSD in the first month of trauma, in fact, if you look at people's brain activation at about 2 weeks after the trauma, you see, and you're looking at, say, fearful faces, a probe that activates amygdala activation to fearful cues in the early weeks after trauma is a strong predictor of PTSD symptoms 3 months and 12 months later. So you're already activating these circuits that later on are associated with chronic PTSD and hyperarousal or hypersensitivity of the threat system. In contrast, we know the hippocampus is critical for providing the safety signals and the safety context signals in the context of threat. And so generally the hippocampus is thought to be a protector of threat symptoms, whereas the amygdala is associated with increased threat symptoms. And we see this as well in the early aftermath of trauma. People who have high hippocampal symptoms, our high hippocampal activation, to an inhibitory task, show very low PTSD symptoms, and people who have low hippocampal activation show very high PTSD symptoms. And again, these are hippocampal probes given about 2 to 3 weeks after the trauma, predicting PTSD 3 months and 6 months later. So we're seeing similar circuits. I talked about the hyperarousal system and the sympathetic nervous system. One of the best ways to measure sympathetic arousal is the galvanic skin response. It's essentially the sweat response, and it measures the sympathetic activation. And if within a couple hours after trauma, often while people were strapped to a bodyboard waiting for their neck spine to be cleared, for example, in a car crash, we would simply ask people, what brought you into the hospital? And the level of their sympathetic response before that question and after that question, as shown here in skin conductance, was a robust predictor of PTSD 6 months later. So again, early hyperactivation of the sympathetic nervous system seems to be one of those biomarkers. So happy to talk more in the discussion, but we're doing this along with predictive analytics and blood-based biomarkers and other things. Well, another component we're looking at is genetic risk. And again, this is one of many studies. And if you follow the genetics field, you know that 2 decades ago, we thought 100 people was a reasonable sample. Then we realized we needed 5,000 and then 20,000. And really, we need on the order of hundreds of thousands of people for large-scale genetic studies of heterogeneous disorders. I was fortunate to be one of the founders of the Psychiatric Genomic Consortium PTSD working group with Kirsten Coman and Caroline Nievergelt. And this group, through over the last 10 years, has really brought worldwide groups together. And we now have over 1 million participants and over 150,000, I think, cases of PTSD. And we really have a genome-wide association map or a Manhattan plot that looks almost as robust as schizophrenia. So we're really starting to have a genetic architecture of how trauma affects the brain to create PTSD-like symptoms. I'm, of course, not going to go through all the individual genes, but what I'll do is point out a couple of them. And this is an intermediate. This was one of the GWASs from Murray Steen and Joel Gartner in the whole Million Veteran Program group, with about a third of the sample size as the whole PTGS, the FREEZE-3 PGC. But most of these clusters that were found with several 100,000 held up robustly at 1 million. And there's many different genes in here, some of which are entirely novel, some of which are associated with other disorders. But interestingly, we see quite a few that are both related to stress and HPA regulation, in particularly the locus, including corticotropin-releasing hormone, which I'll come back to, as well as those as a number of genes involved in synaptic plasticity, whether it be CAM kinase or phosphatases, orexin, and others. And so the theme of plasticity and the theme of stress signaling is starting to be validated at the large-scale genetic level as well. So I'll diverge for a minute and talk about the hypothalamic pituitary adrenal axis. And again, this is the longest understood endocrine component of stress. And when the amygdala is activated, it activates CRH release in the amygdala, in the bed nucleus of the stria terminalis. That activates CRH release in the hypothalamus to the pituitary gland, activating ACTH, activation of the cortisol and adrenaline release from the adrenal gland. And that's the classic system. But it's also been studied for a long time related to PTSD. So it was really great validation that the CRH receptor, the main target receptor of this, is also validated at a large-scale genetic level. Just a few studies over a couple decades, long before that genetic study, looking at the role of CRH in model systems. This, for example, we showed in the early 2000s that viral vectors that overexpress the CRH receptor, so making more CRH receptor, which is generally associated with more activation of the receptor itself, was associated with increased startle, again, one of those basic components we see with PTSD, and a number of other threat-related symptoms. If we used another genetic approach and made the CRF cells in mice more active, we did that by specifically knocking out the GABA receptor, an inhibitory receptor, on the CRF neurons by crossing CRF Cree mice with the GABA floxed mice, so that we essentially decrease the GABA inhibition of those cells. When we did that and had CRF cell overactivation, interestingly, we didn't see an increase in fear learning itself. This is using tone shock pairing and classic Pavlovian conditioning. But when we tested the animals later, they showed more fear initially, and importantly, they showed a deficit in extinction of fear. They continued to be afraid despite evidence to the contrary that they no longer should be afraid, which is one of the core components of PTSD, and they showed a hyperactivation of the HPA axis. That's some of many studies, and just one other one that I'm not going to go through all of this, but another gene that's been very interesting is called FKBP5. FKBP5 is a gene that regulates the CRH gene itself and regulates CRH receptor, and it's highly sensitive to cortisol. And its main job is actually regulating cortisol feedback on cells and turning off the HPA axis. And what we found early on with Elizabeth Binder, who's now the director of the Max Planck in Munich, when she and I were collaborators at Emory, was that particular polymorphisms of this stress regulating gene were associated with differential sensitivity to child abuse for later developments of PTSD. Those with the resilient, less reactive version of FKBP5 showed not much increase of PTSD despite child abuse. Those with the very sensitive risk allele of FKBP5 showed a very strong relationship of developmental abuse with adult trauma leading to more PTSD. And this same risk allele showing increased HPA axis with trauma also showed hyperactive amygdala association in other studies, and decreased or dysregulated hippocampal functioning in yet other studies, and were associated with gene bioenvironment interaction in large-scale meta-analyses. So all of this leads us to a general model that the prefrontal cortex and the hippocampus are constantly regulating the amygdala. And the amygdala's job is to constantly be pulling in information from the external world and leading to stress responses or resilience responses. The combination of risk factors, child maltreatment, poverty, violence exposure, all of the effects of structural racism and others, combined with biological factors, genetic risk, history of genetic risk, differential HPA axis, may then lead, and in this case, FKBP5 as a modulator of developmental cortisol, lead to a system that is hypersensitive, in such that early life stress leading early cortisol is one example of how you get sensitized amygdala through some of these gene pathways related to amygdala regulation and HPA regulation, so that as an adult, when you experience trauma, you're now more likely to go down this sensitization path and stress and fear path. Okay, so in the last few minutes, I want to switch gears and talk about once you have the threat, once you have the trauma or PTSD, how can we improve it? And I started with this figure here showing that exposure therapy takes high levels of trauma and takes it down to low levels of trauma. And one of the components that we know is critical for learning and memory broadly, and certainly emotional learning and memory, is glutamate activation in the NMDA receptor. And in the NMDA receptor is one of the places in the brain that is most highly expressed is in the basolateral amygdala and the hippocampus. Mike Davis's group initially showed that NMDA receptor learning was critical for extinction of fear. And the basic idea here was this: they took rats, trained them to be afraid of, in this case, of light. So they had rats in a small chamber, they turned the lights off. Every time the light got turned on, they got a foot shock. So it took only about 5 or 10 of those for then the rats to be very afraid of this context when the lights came on. So when they now took these animals and turned the lights on, they showed high levels of fear, either with the acoustic startle reflex or with freezing. They then did extinction. They did psychotherapy for the rat, if you will. They gave the rat 60 lights in the absence of any shocks, and then they tested them again. And now they weren't afraid. That's the extinction signal, and they would stay not afraid over time. If they did that same exposure to the lights, though, in another group, but at the same time they gave them an NMDA blocker or antagonist, either systemically or directly in the amygdala, and then they tested them the next day off of drug, those animals were just afraid as ever. So really important point here. When we do exposure therapy or do extinction, we're not forgetting the memory, we're learning to actively inhibit the memory, and that learning to inhibit that memory is NMDA dependent. So what we did then is took a drug called D-cycloserine, and that is a partial agonist at the glycine-serine modulatory site on NMDA receptors. And it had been shown in the 80s that this makes the NMDA receptor work better and enhance certain forms of plasticity with the NMDA receptor. And so then in 2002, we flipped this experiment around and said, can we make extinction work better by making the NMDA receptor work better? So we trained rats to be afraid. We gave a partial extinction with 30 lights. They were less afraid, but still somewhat afraid. And then we did that same partial extinction with D-cycloserine at the time of extinction to make the learning work better. And in fact, it did. And we had used D-cycloserine because it had already been shown to be safe in humans, and it had gone through a number of trials in other disorders. And the take-home point was, in humans with only 2 rounds of exposure to fears of height, they got much better as if they had normally gotten 6 or 8 sessions in a double-blind, placebo-controlled way. So we could have humans extinguish fear of height in a more rapid way, just like our rats extinguished fear by giving them D-cycloserine. So this was a very exciting time. That was in 2004. And over the next about 5 years, that was replicated and extended in a number of different disorders that use exposure therapy, in social anxiety disorders, across multiple continents, in exposure and response prevention and OCD, in people with PTSD, with more severe PTSD, got better faster with D-cycloserine, and in panic exposure. However, as with often happens with a new idea that's working great, and I'll come back to ketamine in a minute, as more studies kept going, it didn't replicate. And we weren't sure for a while as a field, well, was this because people were getting D-cycloserine from different groups, or were we using different doses, was it different timing, all of which were somewhat true. But one of the most interesting components that we think is now the explanation is, for example, an Adam Gustella's study in Australia. They found that people who got D-cycloserine and got better within session, they got much better over time. But if they got D-cycloserine and did not make within session extinction improvements, they actually got worse or no better. Animal studies at the same time showed, if you remember me talking about reconsolidation with sensitization, that anytime a memory is reactivated, that same engram can either be strengthened or it can be weakened or can be suppressed depending on how much exposure and other components. And a group in England showed that D-cycloserine actually could strengthen, could enhance reconsolidation of fear or extinction of fear, depending on how much exposure they got. The problem is, this is not that easy because every human needs a different level of exposure to be, and so we still don't really know where we are with most people on that curve of either resensitizing versus weakening that memory. But now new studies have started to show with better controls and being able to better control the timing, as well as maybe enhancing other components like suppressing the HPA axis, that D-cycloserine may still be useful. It's a generic drug and probably will never be used as anything other than sort of a research tool, but it's an interesting example. And where I think it's important is now with ketamine. Now, while ketamine is an antagonist to NMDA receptors, it's thought that it actually enhances glutamate plasticity by inhibiting the GABAergic suppressors. And so ketamine effectively is an NMDA-dependent plasticity enhancer. And people like Adriana Fetter and others have shown that if you combine ketamine treatment with exposure therapy, you can get much better. My worry is that whether it be ketamine or BDNF or D-cycloserine or others, anything we're doing, and now there's a lot of excitement about psychedelics, all of these are about enhancing plasticity and combining enhancing plasticity with therapy. But with PTSD at least, if we don't have good control over whether we're driving the memory down a reconsolidation, sensitization process or an extinction process, we might not be able to control it as well as we would like. And so I will end then with where I think the future holds, which is not just general plasticity, but targeted plasticity towards the fear off or fear inhibition pathway. Could we, if we knew the difference in the circuits between reconsolidation and extinction, just drive plasticity and extinction? And this may be possible because it turns out that the amygdala is made up of both fear on and fear off cells. All the cells in the amygdala aren't doing the same thing. And Luthi showed that there's a certain cells in the amygdala that fire to the fear process, to the fear conditioned cue itself, but there are others that don't fire to a fear conditioned cue, but then are activated by the extinguished or safety cue. And while I talked about the amygdala as one thing, we now know that there's about 15 different subnuclei in the amygdala, and there's literally dozens, if not 100 or more, different specific cell types. And so this is where modern molecular biology and cell type specific sequencing is starting to come in with old school circuit biology. And in fact, there's one set of cells in the basolateral amygdala that are marked by the Thy1 developmental gene that seem to be these fear off specific cells, in that they don't seem to be active with fear conditioning, but they seem to be active with fear extinction. And when we activate these cells using optogenetics, a genetic tool for turning cells on and off, we can specifically inhibit that flow through of the amygdala from the sensory information from the external world into the reflexive central medial amygdala. And when we activate these fear off cells, we get a robust inhibition of fear learning and an enhancement of fear extinction. And we do that if we do it with optogenetic tools or with chemogenetic tools, we get the same thing. And in this case, we can essentially completely block fear by activating this specific subset of cells in the amygdala. And so the idea is we can identify genes that are marking these fear-off cells. And this is one case where we found a gene called neurotensin 2 receptor that was specifically expressed in the fear-off population. And when we activate neurotensin receptor with a drug called beta-lactotensin, which is an agonist for it, you get a robust decrease in blockade of fear and an enhancement of fear extinction. And so all of this is to say, and with Keitai at a recent Nature paper showing that neurotensin can switch the valence in the amygdala. So all of this is to say, we think there's a new neuropharmacology coming down the pipeline based on our understanding of the neurobiology of the threat on or fear on and fear off pathways. And if we can start to get those down to the level of prediction that really translates across species, one way we're doing that is sequencing in humans and mice at the single cell level to show that when we're studying a specific cell population in mice, it's the same one that is involved or at least available in humans and differentially activated in PTSD or not. So in summary, PTSD is a combination of things that range from early genetic differential risk, effects of consolidation, and differential expression of symptoms that leads one person down to more of a generalization, sensitization, and fear hyper-responsive state. And another person is able to recover through these other brain systems, discrimination and extinction. By studying consolidation, we can start to understand this better in humans and develop systems where we could do early interventions. By studying large-scale genetics, we can identify pathways that are clearly representative of the biology and then understand their mechanisms. And by learning to enhance extinction, we may have novel ways of enhancing recovery. At the broad level, what happens with PTSD is the amygdala is out of control and essentially the brakes fall off. Our trauma-informed therapies increase top-down regulation. One of the questions in the chat was, what is EMDR? And what we think EMDR is doing is grounding the person, whether it be through eye movement or through tapping or through other distraction. So the person is able to talk about the emotionally difficult content and not dissociate. Dissociation is essentially cognitive avoidance and prevents the extinction from working because your brain goes in a different place. But if you stay grounded, you don't dissociate. Our current medications, which are really just SSRIs, though there's others like prazosin, which are very interesting and I could discuss, which essentially decrease that sympathetic arousal. But for the most part, our current medications just dampen the symptoms. They're not getting at the underlying problem. Our hope for the future is medications combined with therapy that really help the plasticity components in the right circuits work better, faster. And I'm not going to go through all of this, but there's a whole host of components by understanding fear, anxiety, and PTSD, and understanding the conserved neural circuitry and how that may lead to novel targets. The take-home message is a precision medicine approach to trauma and psychiatric disorders may be feasible based on understanding neurogenetics and circuit dysfunction. And I'll just end with thanking primarily NIH funding and a large set of wonderful collaborators over the years, and I try to thank people as we went. So with that, I will stop, and I look forward to discussing.